is we determine what causes the condition, right? We're all going to learn in Module 7 as infectious disease, Module 8 is non-infectious disease, and within a subclass of non-infectious disease, you have your genetic disease, okay? Now, when we're talking about inheritance pattern, what are the different kinds of inheritance that we can have? A bit louder. Yeah, sex-linkage is one of them. What did she say that the last two students didn't say? Good, right? You need to identify the type of dominant group. I'll tell you why that's important. Ethan, how many chromosomes do you have? 46, right? So you have 22 pairs of these chromosomes here. What's different or unique about the 23rd pair? Yeah, they're your sex chromosomes, right? So what does a male have? Yeah, good. X. Why is it called a Y? Who can tell me? In biology, you should ask why a lot, and you'll eventually always get to a really good answer. Doesn't a Y look like a degenerate X? It looks like a redundant or like it's missing or disabled X chromosome. That is what a Y chromosome is. It only carries about 6,000 genes and doesn't carry the same allele as the X chromosome. Okay? Good. So when you guys are telling me dominant or recessive, that isn't telling me much. You need to tell me first where this gene lies. If the gene lies here, what do we call it? Good. And if the gene lies here, what do we call it? Good. So it's the first word you must tell me. Okay? Now, what does it mean to be dominant, Tashia? To be dominant is to have a gene that won't be expressed, will be expressed. Okay? Does anyone want to be as specific as possible here? Really? What's a gene, Varun? Good, right? So if I draw a line here, you'll agree there is a region or a set of letters, right? That set of letters that codes for a protein is a gene, as you all know. What do we call variations of a gene? Yeah, alleles, right? So this chromosome would have come from one parent, let's say your father. This chromosome would have come from another parent, your mother, right? The variation of that gene on that first chromosome is called allele 1. Variation of the gene on the second chromosome is allele 2. You all know this, right? I just need you to refresh me so I know you know. Okay? Now, who can use the word allele to explain what dominance means? Georgina?
The dominant allele would be the overpowering allele that would pass the allele.
Overpowering. It sounds like an English word. And English words are fluffy, right? I need some concrete terms. Jessica. Okay. Now, you'll agree there's a dominant allele and a recessive allele, but you're struggling to articulate to me what they do, right? So let me ask you this. The dominant allele we typically denote with a capital R or a capital T or a letter. The recessive, we give it a little one, right? What do we call the genotype when we have a dominant and recessive allele in an individual? Good. Now, which allele gets expressed? Yeah, right. So a dominant condition is one that is always expressed in a heterozygous individual, right? So that's how you can define a dominant condition. Now, I'm glad you all understand this. Now, last lesson you learned how to break down a pedigree, right? So we'll start with this. What does this kind of relationship indicate to you, Georgina?
married couple.
Yeah, very good. So when two individuals have a line that connects them, hopefully you never see this, okay? You're going to tell me why that's bad. They're related and married, right? So you would never see that in HSC Biology, but it happens in real life. Going back to this is a marriage line, the horizontal lines, right? What does a circle and the square represent? Good, right? So typically a circle represents a female, a square represents a male. What does black represent? Affected. Has the condition. We don't know whether it's dominant, we don't know whether it's recessive. The way we get this information is we take a history. That's it. It's very easy, right? What about these two individuals? How are they related? Good. So you understand how to generally read a pedigree, which is good. Now, in an exam, if you drew this pedigree, what do you need to do to get full marks? Very good. Right, I want you to remember this. A band 5 versus 6 student, the difference is a band 6 student always has a key. Varun, what's a key? Yeah, so just this is female, this is male, and shaded, affected. That's simple. So reading a pedigree is very easy. My question is, how do you solve a pedigree, right? That's the hard part. What's the goal of a pedigree? We're trying to find the inheritance pattern of a disease. Now, you all just told me there's 4 different types, 4 main types, right? There's autosomal versions of dominance and recessive, and there's sex-linked versions of dominance and recessive. For now, we won't talk about the other 2 that I haven't mentioned. What other 2 haven't I mentioned? I'll help you out. Rowan cows, snapdragon flowers. Does that mean anything to you?
Like it's kind of like a blend of the, it's not like a or it's dominant or recessive, it's kind of like a blend of the.
Good. I'm looking for a name. And? Good, right? So that's very rare. But I want you to think about it right now. In 1 second, looking at a pedigree, how would you be able to tell that it's codominant or incomplete? Keep it in the back of your mind, because I'm going to ask some of you soon. Good. How do I do this? I'm stuck. First step, Ethan, what do you do? At the end of this class, you should all be able to solve a pedigree in 4 seconds. That's the speed in your head, okay? We'll see if you can do that. Go.
Don't you have to choose which one you want to try?
Okay. Now, if you follow that rule, you will solve this question in 5 minutes. Or maybe 1 minute if you're lucky. If you pick the right one first, right? So what's a faster way? Yeah. Good, I like that, right? Why is Varun checking if males or females are affected in particular? Very good. Now, this is where experience comes into play. When you have a sex-linked condition, what does sex-linked recessive and dominant mean, by the way? I don't want to confuse people. What does sex-linked dominant mean? What does sex-linked recessive mean? We'll use a female as an example and a male as an example. So for a sex-linked dominant and X-linked recessive condition, what does it mean? Georgina.
The disease or the illness or the dominant allele carries it and for recessive.
Yeah, very good. So when you see the word dominant, all that means is capital R equals disease. The dominant allele is disease. When it's sex-linked recessive, it's the little r equals disease, right? X-linked just tells you it's on these chromosomes only, the X chromosome. That's that easy, right? So let's think about this. In an X-linked dominant condition, if a female has even 1 capital R, what does she have? Disease, no disease? Disease, right? Because even if she gets a healthy little r, that dominant will power, aka fully express itself. Okay? What about in an X-linked recessive condition? If she has 1 capital R, does she have disease? No, because the little r, or the recessive allele, is disease. What if she has diseased allele now? It's good. It's masked out. Okay, so she's a carrier, but she doesn't actually express the condition. Very good. What about with males? If you're X-linked dominant, if a male gets a little r, what does that mean? Yeah, he doesn't have the disease. Very good. But if he gets a capital I here, what does that mean? He has a disease. Very good, right? Good. So now that you generally understand X-linked dominant recessive, very good point, Guru. Now tell me why you're looking for males or females. What does it mean when more males are affected? What does it mean with the type of inheritance? You can straight away tell me. Sex-linked? No? Good. I want you to remember this. If all males are affected, it is X-linked recessive. The reason is, how many X's do you see there? One, right? So if alleles are only on X chromosomes, how many alleles can a male inherit? One, right? So there's only one spot here. Everyone agree? Now, if this male gets a little R, he's got disease. He can't even mask it. Whereas a female, she has like an insurance policy, right? If she gets a little R, this is literally a biochemical insurance policy. She can go, oh, I don't know if I really want that. I'm going to play the lottery again. And if she's lucky enough to get a capital R or a functional healthy allele, that will mask the disease and she won't be affected. So we see this disproportion where more males are affected in an X-linked recessive condition. Does that make sense to everyone? Any questions at all? Good. So if it is mainly males affected, your first bet is X-linked recessive. Everyone with me? Good. What does it mean if all females are affected? Very good. Is that common? No. Now, why do you say that? If all females are affected, it's X-linked dominant. We'll play that game again. Imagine that we have the X-linked dominant condition. Let's say this male inherits a little r. Martin, in an X-linked dominant condition, is it capital R that's disease or little r that's disease? Capital R, right? The dominant allele is a disease one. Good. So Martin, question for you. If a male gets a little r, does he have the disease or does he not? No, he doesn't have the disease. Really good, right? He won. Say a woman gets a little r. And does she have disease? Any ideas? Does she have disease? Yes. No. But what does she need to do again? She needs to play another round of this death lottery, right? If this condition causes death, this is another death lottery. And if she accidentally gets a capital R, what does that mean? She's twice as likely to get a capital R versus a male. Does that make sense? Because she's playing 2 rounds of this inheritance. So in a female predominant condition, you say it is X-linked dominant. Make sense? Good. You will rarely see that. I want you all to put an asterisk next to that. 99% of HSC exam questions will be X-linked recessive. So if you get X-linked dominant as your answer, just double check it. Okay? Very rare to get X-linked dominant conditions. Good. What's the next thing I do? Very good. Look for parents of one color with a child of another color. Now, we'll use this question as our example. Do we see that? Gayatri, I want you to tell me, I know you're online, but you can unmute and answer. Do you see parents of one color and a child of another color? I want everyone else to look as well, because I'm going to ask you as well.
Yeah.
Good. Tell me, which generation are they in? The parents. Are they in generation 1, generation 2, generation 3?
Generation 2 are the parents and generation 3 are the children.
Good. So do you all see the parents? The way you say this is that Gen 2, individual 1 and 2, and then Gen 3, individual 1. Now, what can you all immediately tell me now? What is the inheritance pattern? I want to see who caught on from last person. Very good. Why do you say that? Okay, I want you to remember this. Whenever you have parents of one color and a child of the opposite color, the only way this can happen is if both parents are heterozygous, aka capital R, little r, and the child is homozygous recessive, right? So whenever this happens, just robotically write capital R, little r for parents, and little r for the child. Now, you can think about what this means. What color is the child? Are they affected or unaffected? And what allele do they have? So what does that tell you? The little r equals disease, which means this condition is likely autosomal recessive. Good. Do we see mainly males being affected? We do see a 3-to-1 ratio, but usually the sex-linked condition, we pretty much see all males affected. Do you see this male's not affected? This parent is not affected, so kind of keeps me a little bit away from that. Good. Now, you would have done this in 4 seconds in your head once you've got the template. My question is, you've actually just made a really, really good educated guess. You actually haven't proven it. There's only one way to prove a pedigree, whether it's autosomal recessive or sex-linked dominant. And Ethan mentioned it. What is that one way? This is a final step that you do only if you have time in your exam. You don't need to always do this step. Good. Test and disprove the others. I'm going to give you all, have you all done this question already? No? All right. You'll have a minute to disprove why this is not sex-linked. I'll give you 2. Okay, I'm actually going to split the class in 2 now. So splitting you right in the middle. All of you here disprove why it's not autosomal dominant. All of you here disprove why it's not sex-linked dominant or sex-linked recessive. You guys have a harder job. I'll give you a bit more time. Gayatri, any questions at all? Partial, Lukman?
Yeah, I had a question.
Okay. What question did you have?
So I've done some questions and I've seen that sometimes the males, like most males are affected, but not all of them. So how would I guess, distinguish if it's sex-linked or autosomal? Would it just be by testing and disproving, or?
Lukman, you wouldn't be wrong to guess this could be sex-linked, but then you would follow through with step 3, which is testing and disproving.
Yep.
So I wouldn't blame you if you said, okay, this is probably sex-linked. Let me disprove the other types of inheritance. And then when you disprove the other types of inheritance, you'll kind of notice that it may or may not be sex-linked. But my rule of thumb, Lukman, is I don't look at males versus females. I actually look more at all the males as well. So I look at both those things. Okay? So I can see here that even though there's 3 males to 1 female, not all the males are affected. There's a lot of males that are unaffected, which cues me into maybe it's not sex-linked recessive. Okay? But you're not wrong in your approach either.
Yep, all right.
Thirty more seconds. And you will get your exam results next week because there's a lot of trial students this week, so we're going to go through content for the entirety of this lesson. Does that make sense? You guys, have you sat your? You haven't sat your bio exam? No, so this is for chem class. But I'm just giving you all a formal reminder. Your bio exam will probably be in about 4 weeks. Okay, so we're approaching high class. And who remembers what I said you need to submit to sit your exam? Yeah, so your theory booklets, which will all be marked and checked. And what else? Yeah, the HSC trial question booklet for module 5 that you were given. You'll have that? Did none of you get the trial question booklets? Okay, well good. That would mean that I'll give you an extension to do that, okay? But your theory booklets are the main thing on one-handed, okay? Even this lesson now.
Hi guys, I'm just going to quickly take attendance. Okay, so Alyssa? Yeah. Ethan? Georgina? Yeah. Harshin? Is he online?
Yes.
Jessica? Yeah. Neem? Are you online?
No.
Pranav?
Yeah.
Raya?
Yeah.
Ria? Yeah. Tanisha? Yeah. Tashvia? Yeah. Varun?
Yeah.
Isha? Abhishek?
Yeah.
Krushi? Yeah. Maya? Sachita? Lukman?
Here.
Kirtana? Yeah. Kiara? Sophia? Harshita? Gauri?
No.
Gayatri?
Yeah.
Martin? Jawad?
Not here.
And Bushra. Did I miss anyone? Thank you.
Okay, now, I made a bit of a typo when I wrote the title of this. So, it says module 55 trial questions. So, take one pass. Okay. Here are all the trial questions for all the trial papers up until 2020. And you get 2021 trial questions on the website. Okay. Now, you all have a 10-week time period to complete all of this and hand it back. Okay? Since you got it now, typically what happens is you get the trial questions at the start of the module, and by the end of that 10 weeks, when you sit your exam, you finish your theory book, but you also finish these questions. Okay? Now, I have a very important rule with these trial questions, and it's because it's something that I did that I didn't realize was probably the most helpful thing to doing well in HSC science. And it was, once you do the question, what do you all do after you do a trial question? Let's say you do a question, you just close the book and you're done. That's the study, what do you do? Ashria? Good. You need to mark the question, because you have to realize the HSC exam is 3 hours. No one takes 3 hours to do 100 marks of questions. It's too much time, right? The last half an hour to 1 hour of that exam is you double checking your answers to make sure you've hit every single mark. Now, a good student picks up at least 15 marks in the test, right? The first time I did finish my paper, I wasn't at 98 my mark, I was probably at 80. It was the second round where I picked up all the mistakes I made, changed my MCQs, added points to my long responses. That gets you close to 100. Students that never look at sample answers or marking guidelines have no idea what they did wrong or what they did right. And you carry on those mistakes till the end of the year. So one of the most important things is is when you do the question, while you do it, just open a browser, have the edX website, that paper open, because it tells you which paper those questions came from. Just have the sample answers open. Do the question in time conditions, look at the sample answer, and with a red pen, or maybe even in palm cards, write the mistake you made. I think the best thing you can do is when you're preparing for your Mod 5 exam, which will be sitting, or your Mod 6 exam, come in not with all the theory notes, but with a palm card of all your mistakes, so you don't make them in the exam. And that's how you get better in science. Does that make sense to everyone? Good. All right, let's go. Everyone on the left, let me ask, Prithvi, do you mind telling me, why is this not autosomal dominant?
If you look at Gen,
Everyone, I want you to focus on what she's saying. Yeah.
if you look at Gen 3.1, as parents without homozygous two lowercase r's cannot have an affected capital R lowercase r child or a two capital R child, but then that person's affected.
Yeah, I think I want you all to remember this. Do you see this pattern with two parents, one color child of this opposite color? That is actually the same area we can disprove all the other types of inheritance. Okay? So you can say here precisely, as the parents are one particular phenotype, whereas the child is the opposite phenotype, this implies the parents are heterozygous and the child is homozygous recessive. It is impossible for the child to have a dominant allele. Thus, the condition cannot be autosomal dominant. Okay? Now, do you see there's two levels of difficulty here? Everyone can read a pedigree. Most people can solve the pedigree. Very few people can explain it very clearly. And that's what I want you to focus on. It's two different games here. The MCQs where you pick the inheritance pattern is pretty easy. When you have to explain why you picked that answer, it's a little bit hard. So I want you all to focus on refining that. Okay? Good. But does that make sense, what we mentioned there? Okay, so again, you simply describe the parents are one phenotype, child is the opposite phenotype. And what does this indicate? Indicates that the parents are heterozygous. And what is the child? Homozygous recessive. Okay, thus the child cannot have a dominant allele. And thus being affected involves the recessive allele. Okay? Good. Now, over on the right side. This is a harder job. So maybe I'll start with you, Jessica. How do you test whether a condition is X-linked recessive or X-linked dominant? What do I do? It's a pretty tedious set of steps. So let's say I'm going to check whether this is X-linked dominant. Okay? If it's X-linked dominant, what is disease indicated by? A capital R or a little r allele? Capital R. So I want you all to write that. I always do that. Even to this day, I write capital D equals, oh, not D, capital R equals disease, just so I know. And since I'm looking at a sex-linked condition, what are all males? What's their genotype? XY. So next to every single male, I'm going to write XY. Yeah, it's pretty mindless. I don't have to think too hard. And what will I write next to every single female? Easy. XX, XX. Okay. Now, what does it mean to be an affected male? Because I see some males are affected. What does that mean immediately? They must have what allele? Little r, right? Oh, sorry, this is a dominant condition, so they must have a capital R. So next to every single affected male, I'm going to write capital R. See, you don't have to think. Just like color. You know, there's crosswords or coloring things, you just do it automatically. All right. What about, and I put one here as well. Make sure your Y's look like Y's. Good. Good. Now, what about the females? None of the females are affected except for one. What does it mean to be an affected female in a sex-linked dominant condition, everyone? She has one dominant allele? Otherwise? Do we know what it could be? No, right? It could be a recessive allele, but that dominant allele is masking it and expressing the disease. So we don't know. Now let's go on to the final phenotype we haven't accounted for. unaffected females, what must their genotype be? Double recessive. So I'm just going to mindlessly, next to every single unaffected female, I'm going to write 2 little r's. Make sense to everyone? Yes. Now, where is the issue? Let's have a look at this. Here is one female that has 2 little r's. Really agree? One little r came from the father. Where did the other little r come from? Mother. Next to the mother, I'm going to write a little r. Now, is there any discordancy here? Does anything not make sense? Good. What was my hint to you? Where will you find all the mistakes? In that same area you found your answer. So stay close to that. What do you notice? There is a daughter who has 2 little r alleles. Well, the mom can only give 1. That means a father must be able to give another little r allele. But can he? So what have you disproved? Sex-linkage dominant. So that is how you disprove a type of inheritance. I was explaining things, so I was slow. It would take about 1.5 minutes, which is why I'm telling you. Usually skip this unless you have time. Okay? Now, questions? Anyone? Yeah.
How do we know that the X will it be recessive because you compared to?
Oh yes. Apologies, that was my bad. So all the capital, there should be a little r. Good. So as we said, the affected males have a capital R. So that was my mistake. The male should have a little r here. Okay. Now, same thing. Do you see the error here? Yeah, just it's with person 1. The parents only have little r's. How can one of the children get a dominant allele, right? Does that make sense? Good. Good. Good pickup. Now, I think recessive. How are we going to prove or disprove that? We're going to do the same thing. I want you all to do it yourselves now. Those of you who have it, follow these exact same steps, but with sex-linked recessive. And after this, we might do maybe one more pedigree question, and I'm going to move on. Does anyone know what schizophrenia is? Yeah?
When you hallucinate or like forgetting?
What kind of hallucinations? Do I see an imaginary class of kids? What is a hallucination if I was schizophrenic? It's usually auditory. They hear voices, right? Pretty scary. If you see them in the emergency department and they're having an episode, you know, they can talk to the air. It's quite frightening. Okay, what else do schizophrenic patients have? What are the symptoms of signs? Delusions as well. Now, the reason I'm bringing this up in this context is what percentage of that do you think is genetic versus environmental? Schizophrenia is triggered by many things. Someone can have a really depressive episode or PTSD that can put them into it. Some of you definitely can get it, just a matter of circumstance. But how much of it is genetic? Because we can map genetic conditions like this. Guesses? 50% of schizophrenia is genetic, right? 50%. So we have no clue who has it or who doesn't. Some people in a population just have the predisposition. And then when you're faced with maybe you take something like cannabis, it actually can put someone into schizophrenia. So yeah, it's quite interesting. Okay, let's start. I am going to get Alicia. You give it a go. What do I do? First of all, tell me which one equals disease, capital R or little r? We're talking about sex-linked recessive. You all need to follow these steps. Little r, very good, right? Okay, now next to all females, what are we going to put? XX, right? All females get XX. And for males, what are we going to put? Good. So I'm just going to do X, Y, X, Y, X, Y, X, Y, X, Y, X, Y. What do I do next? Effective females, what do I do? Okay, good. All effective females, does everyone agree, must have two little r's, right? So good. So just blindly, next to all effective females, there's just one, just put little r, little r. What about effective males?
It's just the big one.
Capital is
Affected males. Little r, very good. So affected males will all have one little r. Good. What about unaffected males? Good. So we're just going to find all the unaffected males and put a capital R. What about unaffected females? What must they have? Yeah, so at least one capital R. We don't know what the other allele is. It could be a recessive allele that's being masked by that dominant allele. So I'm done. Now, where's the issue? Like, we can start filling things in. One parent has a capital R only. The other parent has two little r's. So what must this woman have? She's got this capital R from her father. Do we all agree? Because the mother doesn't have any capital R's. So other little r must have come from mum. Good. What about this person? Same thing? Good. Now tell me, where is the issue? Gen 3. Do we see any issue? Georgina? Yes? No?
One, it should have a dominant R instead of the recessive, because the mother has the.
But the mum has, I want you all to pay attention to this, the mum has a capital R and a little r. So when the Y is passed on to the son, the X comes from mum, right? The mum could have passed on her little r, this one right here. That works out. What about here? This capital R could have come from either parent, right? It could have come from the dad. Does everyone see the capital R in the dad? So the mum could have passed on her little r, or her capital R. So it doesn't matter. Anything here. So could we fault this pedigree at all? So what does that mean? There's actually two possible types of inheritance. And this can happen. It won't happen in your exams. They'll make sure they pick an example where it doesn't happen. But that's the limitation of a pedigree, right? You see, in 4 seconds we came to one answer, but only when we tried to test and disprove did we realize there is a whole other option that is equally true. One is not more correct than the other. We just don't have enough info. Does that make sense to all of us? Good.
So when it says like in the line there, I see there's no have to recessive value. But can we see that?
Yeah, see, very good. They're helping. I2 does not have the recessive allele. We already found that though here. If it was autosomal, yeah, see, we didn't use this piece of information because I just wanted to go through the pedigree. But this should help you differentiate between one or the other. Yeah, so you guys can redo this question with that piece of information. But I wanted to run through this just to show you how to break down a pedigree. Does that make sense to everyone? Do you all want to do one more pedigree or do you want to move on? One more? Okay. For this one, I'm not going to help you at all. You're going to come to an answer, and I'm going to ask all of you to see how your pedigree analysis skills are. Sure, give me 1 second. Let me try to find it. Okay. This is from my HSC paper, 2016. Any questions over your house?
Yeah.
Have you done Pedigrees at school?
Yeah.
Okay. Okay, good. Yeah, it's kind of similar, right? So it's probably not sex-linkage. Exactly, right? So we'd say it's probably not sex-linkage. What was the second step? Two parents of one color, a child of one color. Maybe the two parents of one color. What are the child? Good. Now, what did I tell you? The genotype that parents must be. Good. So just write that down next to parents. It's like you're going to capital. About X's, when you say X, that means it's sex. We need to write X's and Y's. So those are the sex conditions. What's the That's all digital disease, right? The child's affected. So what kind of inheritance? Good. So what's the sex? So you've got one option. Now, you need to test that. That's the hard part. Okay. Yes, question. Do you agree you're excessive, by the way? Okay. Now, what was the first step that you did? The first step is actually excessive. Because females are affected. So is that similar? Yeah, right. So it's probably not sexy. So it's either dominant or recessive or so. What's the second step? What's the pattern? See the pattern of your parents being affected then. Is that what you did is that you assumed the type of inheritance was affected. What do you mean from that? What does that matter? Your parents are one color, child of the other color. Where do you see it?
And they have a
So what can you tell me now? They must be.
and then child
What kind of genotype must the parents Maybe writing a line just in case. And then what was the topic? I'll do it. You're right, X is Y, so it's not sex. And it's Dominant or recessive. Capital R disease. That's capital R disease. That's dominant. This R equals disease is. There you go. So what's your answer? I think that's good. Yeah, good. Does that make sense to you? How's the iPad? Good for notes and everything? Cool. What are you doing? You can actually search, so if you write stuff, you can actually search it, and find every page you wrote that word. If you want to search Pedigree, find all the pages you had in Pedigree. Yeah, I like that word. Alright, let's get started. Alright, I'm going to ask one person. I want everyone else to listen to that one person. Okay? Now, we'll start with the exam breakdown, right? So don't worry about explaining how that happened. I want you to tell me your steps you followed to come to a quick, educated guess on the inheritance pattern of this pedigree. Okay? There's 3 steps I gave you. Okay? Let me ask Ethan, 3 steps as fast as you can.
The first one, there's no technique.
Oh?
Because it needs to introduce how.
Good. So males is relatively similar to females. So it's probably not second. Good. So you're not going to write X's and Y's just yet. Good. Next.
I just saw the 2 parents that are affected.
One child that's affected. Good. Is that the one you found?
Yes.
Good. So what do I immediately write for the genotype of the parents? Good. And the child? Now, little r equals disease, because a child is shaded. So what kind of inheritance is that? How easy was that, right? Already come to an answer. And that's most likely what the actual answer is. So he's made a really quick, educated guess. Now, what I usually do is, this is where experience comes into play. If it's autosomal recessive, what else could it be? Just from the previous question you just did. What else looks similar to autosomal recessive? Sex-linked recessive. So the only other thing I would do is that disprove sex-linked recessive and move on. Right? Very quickly, in 2 seconds, why can it not be autosomal dominant? Because of this exact same rule. Did you all see that? So what that means is whenever you get autosomal dominance as one of your educated guesses, it can never be autosomal recessive. If you ever get autosomal recessive, it can never be autosomal dominant. So the only thing you need to do, if you find autosomal recessive, is disprove sex-linked recessive and then move on. So how about we do that? So now I'm going to ask Ria, disprove sex-linked recessive to me. What do I need to do? Tell me word for word what I need to write. To disprove sex-linked recessive. That is fair. You probably would still get the answer right, but to be very meticulous.
The second thing about this, he has 2 sex chromosomes. That must mean that the father also has recessive X chromosome, which means he would be affected by this.
Okay, good. You've explained it very good. But I want someone to tell me, pen to paper, what I would write as a student to know. Good, right? So first thing I actually do is I write, if I'm proving X-linked recessive, I just write to myself, little r is disease. Very simple thing to write, but I just like to have that as my reference point so I don't make silly mistakes. Next thing I do is all males, I'll write X, Y, right? X, Y, X, Y. And all females, what will I write? Yeah, so very quickly, you can do that. You can erase me. X, 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 X. Okay. Now I'm done. Now, if you're an affected male, I'm just picking random phenotypes at a time, right? So if I'm an affected male, what does that mean? I have a little r. Good. So now with every affected male, I'm going to go and write a little r. Little r, little r, little r. What does it mean if I'm an unaffected male? Good. So next to every unaffected male, I'm going to write capital R. Capital R. Now, next to every affected female, what am I going to write? Good, right? So every black circle, I'm going to just blindly write little r. Now, for every unaffected female, what am I going to write? Oh, I missed. Second generation. Yes, thank you. Good. All right. Now, unaffected females, what will I write? One capital R. Very good. Right? They could still have a recessive allele, right? But they don't express it. So if we don't know that, we're just going to write one capital R and a dash, because we don't know. Capital R dash, capital R dash. So that took me about a minute to fill all of that out. Now, where does this all go wrong? Well, we can start deducing things, right? This female has received a capital R from the father, so the little r must come from. Good. We need to start filling out all the other missing points, right? This female has got one capital R, which must have come from mum. Where must the little r have come from? Sorry, where must the other x have come from? From the dad, right? The dad only has a little r to provide. Do you all agree? So she must get a little r here. Okay. Now, if she has a capital R and a little r, does that work out? It's a sex-linked recessive condition, remember? Yeah, works out, because a dominant healthy allele will mask it. That's why I always write this reference myself. Good. So I've pretty much got everyone's genotype except this mum's. Can I get this mum's genotype? Not enough information. Yeah. She could have a capital R, she could have a little r. Doesn't matter. So we're just going to put question mark because it does matter. Where is there a discordance, if any? Start looking around. And what did I tell you? Mainly have a look at this same area, and you might find your answer. Does everyone see the issue? Where is the issue?
Father has a dominant allele.
Yeah, whereas one of the children has two recessive alleles. Is that possible? No. So you've disproved it, right? So hopefully now you're seeing the little shortcuts for pedigrees. You can solve any pedigree in like 4 seconds. The only slightly challenging part is disproving the other similar pattern of inheritance. Does that make sense? Very good. Now, final task. So I know and I'm confident you're all good with pedigrees. How do you explain this in an exam setting? The question was, explain why this condition cannot be sex-linked recessive. Condition cannot be sex-linked recessive. Now, online students, I want you to type your answer and add it in the chat box. So Gayatri, Lukman, and Harshal, I want you to type your answer, and I'm going to have a look at that. And Raya as well. And for all of you in center, I'll ask 2 people at random to give me their explanation. And I want the rest of you to mark them. So this will be say 2 marks. Who's sitting in the UCAT this year? Good. Who's sitting LAT for UNSW law? Anyone interested? Who's interested in law here? Okay. And what about STEM subjects like engineering? Okay, is anyone interested in something that's non-traditional? Marketing, architecture, design? No?
I think construction management.
That's cool. No, I think that's cool. Yeah. Why construction management? You know too many engineers, so you can use the network. I like that. Playing chess. Okay, so you should finish up your answer. Okay. Two marks, 2 lines. Okay, so don't write too much. Could I get Tashfir? Get your answer.
This inheritance cannot be extinct recessive since Gen 2, parent 3, and Gen 2, parent 4 are both unaffected carrying the dominant allele, but they have an affected child carrying a recessive allele. This is
Yeah, it's okay. I want everyone to tell me what. It was a good answer. It was a good answer. But I'm being very nitpicky, right? Anything that anyone can provide as constructive feedback?
I think at the end, specify that the father can't give, can only give the dominant allele.
Yeah. So you mentioned that the parents, each parent has a dominant allele such that they can't give a recessive allele. The issue here is not providing one recessive allele, because one parent can do that. The issue is with a child getting 2 recessive alleles. You need to really specify that. Someone ask one more person. Could I get Alicia? What was your answer?
To be X-recessive, the second generation can carry the dominant allele R. However, if the disease is X-recessive, the child must have 2 little R alleles. The father only has a dominant allele, therefore the disease is not X-recessive.
Good. Did you see how she identified? So pretty much what you mentioned, right? Very good. I can give you all my just ballpark explanation, which is if this condition is X-linked recessive, the Gen 2 parent 3 and Gen 2 parent 4 must have the genotype, and I'll write the genotypes because you can do that, right? Respectively. As a result, they collectively only carry 1 recessive allele. For Gen 3 child 4 to be affected, they must inherit 2 recessive alleles, which is impossible. Full stop. Make sense? Good. So do you all see how I was very specific with why it cannot be the case? So I just liked your answer. That was very good. Okay? Good job. Any questions at all? Good. I think enough pedigrees. You have all the skill and arsenal now to do any pedigree question. It's now in your hands to repeat and practice again and again. Okay? Good. All right. This is definitely not the hardest part of HSC Bio. Now, I want you all to tell me what 2 kind of inheritance patterns this could be. I want you to think before giving me an answer because I wouldn't give you an easy question. Give me two types of inheritance this pedigree could be. Or give me one. Alright. Can you give me one? Very good. What was the second? There you go. So, remember the question I asked you earlier? Just by looking at a pedigree in 1 second, how can you tell if it's incomplete or codominant? The answer is, instead of just having unaffected and affected, you will have 3 phenotypes. In incomplete dominance, it'll be unaffected, affected, and blended. In codominance, it'll be unaffected, affected, and then a mix of both, right? Can you remind me the difference between codominance and incomplete dominance? The answer is what happens in a heterozygous individual who has both alleles. In incomplete dominance, there is a blending. So if it was a red allele, white allele, heterozygous organism would be pink. But with codominance, what happens with a heterozygous individual? Yeah, so there'll be a complete expression in partial areas of that same organism, of one allele, and then complete expression of the other allele in that same organism. In humans, what is the example? Codominance. It is so important because if we don't get this right at hospital, a lot of people will die. I don't know too much about whether it's codominant or incomplete. And the reason because those things don't kill you, and I don't need to know things that don't kill you. Yes, very good. A-B-O blood system is codominant, where the A and B alleles are codominant, and the O allele is recessive. So if I have AB is my blood type, what that means is my red blood cells, who can remind me? Every one of my red blood cells, these biconcave discs, will have A carbohydrates and B carbohydrates. They're not actually A and B, it's simplified for us to understand, but they're different chains of sugars, pretty much. Okay. Good. All right, let's move on. Okay, now just a very quick mention here. You're learning binary traits, right? You learned that pea plants, you learned that they can be tall, they can be short. These can be affected or they can be unaffected. But what about conditions like height, where it's clearly not the case where everyone's either LeBron James or a midget, right? So why is that the case where we have a spectrum of variation for a particular trait? Right here. What is the case? What does polygenic mean? Yeah, very good, right? So in reality, you are learning in HSC Biology there is one gene that makes one protein that can code an entire phenotype, but that's not the case. The reality is there is one gene that codes for one. What do I use? What does one gene code for, everyone? No, an allele in a gene is like saying brown hair codes for hair. It doesn't make sense. One gene codes for one polypeptide, is what I want to hear. Right? This is a whole inquiry question. This is the point we're trying to get across. You can't forget that. And what does that one polypeptide? Yeah, it makes part of a protein that then codes for. This is what you're all learning, but in reality, there's gene 1 and there's also gene 2, there's also gene 3 that also make another polypeptide that will also be part of another protein that will also affect phenotype, etc., etc. So in reality, there are many genes all making different polypeptides that are all making different proteins that interact with each other to code for your traits, like your height, your skin color, your maybe skin oil production, your coarseness of your hair, your freckles or non-freckles, is all in this manner. So when this happens, instead of seeing a binary where you see tall or short, what do you end up getting? Okay. When you all sit your exams, what would you see for HC results on ATARs? See a bell curve, right? Because that tells you that the effort or the whatever genetic factors that were at play were polygenic. Okay, there are many genes that work here. Very good. What about this word here? What's the difference between polygenic and this word? Literally in the word. Yes. Thank you. Jessica? Yeah, so good. I know you all understood multi-allelic means multiple alleles, but I want you to put it into this picture, right? We said polygenic is when there are many genes that are coding for their own polypeptides, that are coding for their own proteins that are affecting phenotype. Multi-allelic states that for that gene, instead of just having allele 1 and allele 2, there's also allele 3 and allele 4, and that makes it even more varied. Also contributes to this bell curve of characteristics. In reality, everyone, there's not just 2 alleles for a gene. There can be 7 different variations of one gene. Right? The fact you're all learning it this way is because the brain can't comprehend how complicated our interior design is. Right? In reality, it looks like this. Does that make sense to everyone? There are more than 2 alleles that code for a gene in reality. There are more than 1 gene that codes for 1 phenotype. Good. Is that the case for pea plants? Are pea plants like this? No, which is why Mendel studied them. This is way too complex to study. Mendel picked pea plants because they only have 1 gene, codes for 1 protein, that codes for 1 trait like height. So it's very easy to study that stuff. Okay? Good. All right, next question. Any questions at all so far? Okay, we've hit the hour mark. What I'll do is I'll give you all a 5-minute break. Stand up, stretch. You can walk around. It's not raining, you can walk outside. But we'll recap at about 12:26. Okay? Good. Lookman, did you have a question? No. Okay. Hi, this is your...
I just had a question for like the demonstration.
Yeah.
So,
Tell me a bit about your assessment first.
so it's just a polypeptide synthesis model, and I finished my model.
Okay.
And I was just looking through, like, so basically we have to make amino acids on histidine and then GIN and tyrosine.
You have to make those, that chain.
Yeah, that chain. So I just wanted to double check my DNA sequence, which made my mRNA story.
Okay.
I don't know if it's correct, but I think it is correct because I double checked with my teacher. I think it was correct, but I just wanted to double check with you before I
The
start.
way you can find that is you should do this is what you would need. So you would have seen this before. Read the code. We should make sure we pick one that's MI.
Yeah.
Sickle. This for a second. Okay, I'm going to assume this is an mRNA table because it usually is.
Yeah. I think I got that correct.
Okay, so you've got the mRNA strand for
Yeah.
CAU.
Yeah.
Good. Okay, so I'm going to assume you got all of that right.
Yeah.
Now, from the mRNA to DNA, you need to go complement.
Yeah.
So you did that. So
Yeah.
CAU should be G T
Yeah.
A. We've got T here. The first letter is C should be
Like, okay, so like on here, this is my strand. So like C is complementary to G, A is T, but then I like the bottom strand, which is the template strand, is what like it's complementary. So like G is complementary to like C, which I made the mRNA strand on. Does that make sense? Yeah. And it's like CA. I got that wrong, but I fixed it. So it's like CAU.
Yeah.
And then it like instead of, because this was my original one, but I just double checked it yesterday. Instead of C, it was G.
Let's have a look. GA. Is it glutamine?
Yeah, it's GIN.
GIN is not. GIN, I think, is glycine.
Yeah.
Oh no, it's a GLN. That's. Oh no, you've got GIN.
I don't know if it's GLN or
That one doesn't exist.
yeah, on the that it just shows up as GLN. I
No,
think
you
they
need to ask your teacher. That doesn't exist. Because the shorthand should be GLN for glutamine, or it should be GLY for glycine. So you need to ask them which one they're referring to.
So is it GLN
which is glutamine.
or GYN?
Yeah, because this is like an international code.
Yeah, because that's why I was getting confused. That's why I had to do mRNA.
But I think otherwise you're following the exact same
Yeah.
steps. So you need to use this table.
Yeah.
That's the mRNA,
Yeah.
which is the complement of the
Yeah. And with the start coding for translation, I know it's usually AUG, but that codes for something else. But should I just have it there to like state?
Well, it starts.
Yeah.
AUG. So
It usually like initiates the translation process.
that's right. The
But
first.
I think I should make a comment on that just to make sure the markers know it is AUG. It does start the process.
Yes, the finding is the first amino acid.
Should I do that yet?
I think.
Doesn't really say, but I think it's just it's good to have background information just to like.
Excuse me, sir.
Yep.
If you don't mind, could you please explain to me that pedigree question, the one with the incomplete dominance? Because I tried to do that at home, but I couldn't.
Sure. I'll explain it in just a moment.
Yeah, no worries.
Okay. So methionine, you're right, is the first amino acid made in translation. That's usually spliced off or cut off in a lot of proteins.
Yeah.
So if you're using the polypeptide synthesis of, okay, do you have splicing in your model?
Yes, I do.
Perfect.
I'll show you my model. The section of splicing, and then yeah, here's the splicing.
And then like the poly, yeah. Like the capping and the tailing, and the splicing of the exons and the introns and stuff like that.
That's
And
perfect.
Yeah.
I think the only thing is, do you have methionine at the end of pre-mRNA?
Should I have that?
I think you should.
Okay. Let me just write that. So add methionine at the end of it.
You don't need to. But if you're going to add the sequence for methionine in front of the strand in your
would that change the sequence of it?
I would say just forget about it.
No.
I don't think they're on that.
All good.
Yeah.
And the stop codon is UAA. Cool.
There's 3 different
Yeah.
stop codons.
So it doesn't really matter.
Doesn't really matter. Yeah.
I think that's all I wanted to ask.
If you would like, if you're going to include stop codons, that means you're going to add to the end of the DNA strand.
Yeah.
Is that right?
I did add it to me.
If you're going to do that, then you should add a methionine at the start of the gene as well.
Okay.
And then you should show the methionine and the stop codon,
So
and then you should slice them off. Making the mature protein.
Okay, so add methionine.
Sequence, yeah.
Oh, does it have a whole separate sequence?
Methionine is just
the 3
it's just
AUG.
AUG.
Okay. Add that AUG.
For the mRNA, which means
Yeah.
DNA will be the complement.
So add that at the mature mRNA.
No, you remove that because so yeah, you'd add that in the mature.
mRNA.
No, not mature
The
mRNA.
pre?
The pre-mRNA, and that gets spliced out by the splices.
And then it gets sliced out. And then it just starts with like CAU. So I don't have to like state that in translation.
Yes, you don't need to show the AUG.
Okay.
You know.
But that's what I did here. I did show. Yeah. I did add that. I added the AUG.
No, actually, show the methionine in the mature mRNA.
Okay.
So the methionine will not get spliced. The methionine will get removed after translation. So it's called a step called post-translation modification. So I would say if you don't want to have all that, just don't even use it. In reality, what happens is there will be a AUG in the mRNA. It will travel to the ribosome.
Yeah.
The tRNA will place methionine at the start of the polypeptide chain, and after it leaves the ribosome, enzymes will break down the methionine and remove it. So I'd say just ignore it.
And I just wanted to double check the tRNA and ones.
Yeah.
So I can show it
Sorry, could you read it?
I can show you off the card.
Yeah,
I'll just show you. Yeah,
after
all
you-
good, all good. Okay, thank you.
Oh, you did?
Look, man, I'll explain the inheritance after class. Is that okay? Just stay back after class.
Yeah, no worries.
Remind me. In the interest of time. Okay, now this is a very important dot point because it often gets overlooked when being taught in class, and there's a lot of exam questions about this, but it's very easy to understand. And the topic that I want you to have, or the title that I want you to have, is calculating allele frequencies. Calculating allele frequencies. Now, to understand how to calculate allele frequencies, you need to know why it's called a gene pool. Why do we call all of the genes in a population the gene pool? Pardon? Yeah? But why pool? We call it a gene planet if it's a lot, right? Why a gene pool? It's because when you have a population, right? Let's say individual is a capital R, another individual is little r. When they have an offspring, what happens? There is one random allele that flows into the organism and another random allele that flows into an organism. So do you see, pretty much every single human is kind of like a vessel that carries two alleles, and that flows to another individual when you give birth to an offspring, right? And so do you see how these letters are freely moving in the population over time, right? The allele gets inherited by one organism, and then it can flow into another organism, or it can be discontinued, okay? So to represent that, we state the word gene pool. In reality, I think it should be called allele pool, right? So everyone in a population is kind of just a pool of alleles, and when you have offspring, the alleles will combine, right? Pick a genotype. Any. Pick a genotype. Oh, same letter.
Like lowercase.
Okay, lowercase, right? So if these organisms have a child, one allele randomly, the capital R will be inherited, and one allele, the little r, will be inherited. Does that make sense? And that's going to change the frequency or the proportion of the capital R and little r allele in the population as well. So we need to start calculating allele frequencies. So now that we've mentioned all of this, you're looking at questions like this, right? Have a read of this question. Cystic fibrosis, a genetic condition where a single letter change in the DNA code makes someone suffocate from the inside with mucus in their lungs. Now, this is the most common genetic condition on the planet. And 1 in 25 Caucasian people, you all know 25 Caucasian people, right? One of them has the mutated allele for that gene. It's very common. Coming back to this, the question here is determine the allele frequency, right, for the cystic fibrosis allele. Any idea how we go about this? Any of our mathematicians? What is a cystic fibrosis allele, everyone? Is it the capital R or the little r allele? Good. They've told you it's an autosomal recessive disease. So little r equals disease. Does that make sense? Right? This question is asking you, what is the allele frequency of the little r allele in the population? Now, how are we going to find that out? Not lowercase tt. See, this is the misconception you tend to have. But I really like your reasoning. But again, I want you all to think of this like alphabet soup. A gene pool is just a soup of letters. Does that make sense? What they are asking you is how many little t's do we have in our alphabet soup? That's literally what the question is asking you. Let's try finding this out. They've told you that for homozygous dominant individuals, there's 634,000 people. So how many capital T letters are there? If I gave you one set of 2 capital T's, how many T's do you have? 2. If I gave you 634,000 sets of 2 T's, how many T's do you have? Twice the amount. So the amount of T, we're going to do a little table here, capital T, and we'll write lowercase t. The amount of capital T you got there is 2 times that. So that's, what is that, 1? Someone quickly do the maths. 12, 6, 8, 3, 0. Good. We've got that many big T's in this alphabet soup. Does that make sense to everyone? Good. Now, we have 96,000 people who have 1 capital T and 1 little t. Right? If I gave you 1 blue pen, 1 black pen, you only have 1 blue and black pen. But if I give you 5 sets of a blue pen and a black pen, how many black pens do you have? 5, right? It's a very simple question, but the reason I'm asking you is, in this case here, Tashmiya, how many capital T's do you have? 96,000 people each have one big T, one small t, right? So how many big T's do we have? 96,000. Do you all agree? Right? How many little t's do we have? 96,000. Does that make sense to everyone? Is everyone with me so far? Now, we have 5,457 people with two little t's, right? Each person has two little t's. We have 5,000 of them. So how many little t's do we have? Times it by 2, yeah. What is it, 10,000? 10,914. Good, very good. Now what are you going to do? We want to find our alphabet soup. What are we going to do? We're going to add all the big T's, add all the little t's, add them together. So how many big T's do we have in total, everyone? Someone get a calculator and do it, because we need one for this. Everyone? Big T's? 1,364,000. Okay, and what about little t's? 199? 14?
This is 100,000.
Q. 100,914. Okay. Good. Now, what is our total letters? We have to add these two together, right? So mathematically, let me put it for you. I put it to you verbally. But what we are truly trying to find is trying to find the number of times that we have a little t in the population divided by the total number of big T's in the population plus the total number of little t's in the population. That is our sample space. Does everyone agree? So the allele frequency of the T allele is equal to this. Right? So how many times has the little T allele appeared? It's appeared 100,914 times. How many times has any letter, big T or little t, appeared? 1, 3, 6, 4, 0, 0, 0, plus 100,914. What do we get as our percentage?
Zero point zero
0.072, convert that to a percentage. Good. They don't care about sig figs or decimal places. As long as you 2 decimal places, you'll be fine. Okay? Good. Now, I want you to just step back and realize what you just did. To find allele frequencies, it's not about finding genotypes. A big misconception students will do for these kinds of questions is they'll find the number of times someone has a homozygous recessive genotype and divide that by all genotypes. That's complicating things. Treat it like a gene pool. Find the total number of letters in that gene pool. That's your sample space. And find the total number of times either the dominant allele, the big T, or the recessive allele, the little t appears. That's it. Does that make sense? A lot of students make mistakes in this because they just didn't learn it at school, but it comes up in your trial papers. Does that make sense? Any questions at all? Good. Online students, any questions at all?
Nope.
Good. Right. This question's a bit crazy. We'll not do that right now, but do that at home. We'll move on. This is a really good question, but I just want to know, I just want you all to think about this. You don't have to write an answer. Think and answer it to me. Let me just see if there's any table. I want you to predict whether, who has greater prevalence of sickle cell disease, Africa or Europe, and why? This is a real life case.
What's the question?
Can you account for the differing sickle cell allele frequencies in Africa and Europe?
What was the question?
Quite literally this. Where do you think sickle cell disease would be more frequent? In Africa, Europe, and why? Ethan? Okay, so Tanisha, you said Europe. You're saying it's going to be more prevalent in Europe. Are you sure? This isn't even biology, it's a thinking test. Who can problem solve? That's what this is. No? So you're saying that sickle cell will be more prevalent in Europe? Does everyone agree? Disagree? Tashvia? Not sure. Any ideas? You agree with Europe? Okay. So let's try understanding this, right? I don't want to spoon feed you all. I want you all to think. Malaria is bad. Right? We have these two countries. We have Africa and we have Europe. Okay? In Africa, in Africa, malaria is very common. You do it in red because it kills people, right? Now, there is another condition called sickle cell trait, right? And what does it result in? Sickle red blood cells that have reduced susceptibility to the plasmodium, which causes malaria. Okay? So now I want you to imagine there are 10 people with sickle cell trait in Africa, 10 people with sickle cell trait in Europe. Okay? Now, has anyone heard of sickle cell disease? No? No one's heard of it? Okay, it's a trait. Okay. Now, these 10 people have reduced susceptibility to malaria. So will they die from malaria? Maybe not. Less likely to die. Do you all agree? They're less likely to die. What are they more likely to do? Survive, reproduce, and pass it on. So that 10 might become 20. Whereas here, is there any selective pressure acting on that sickle cell trait? Is there anything favoring the survival, not favoring the survival? Well, there might be something not favoring the survival because it's a bad trait to have, but if you didn't know that, that's fine. Just keep it at 10, right? If this repeats over many, many generations, what's going to happen to the prevalence of sickle cell trait? These 20 people, again, less likely to die from malaria, will survive, reproduce, and increase the prevalence to 30. And if you do that over centuries, what do you notice? Sickle cell trait becomes very common in Africa and very uncommon in. This is a real case, right? In the United States, sickle cell disease very markedly affects the African American population, right? And it's much more common in Africa because it actually helps you survive against malaria. Make sense? Good. Did anyone think of that before I explained it to them? Okay. Did you see why that question was a bit challenging? It wasn't just memorizing, it was applying the knowledge. Okay? Good. All right, we're going to move on now to a very interesting topic. Who's learnt about SNPs? Anyone learnt about SNPs before? Not done it at school yet?
We've done the school, but
Didn't go through it at all, but you did it at school.
we just like read it slide.
You read the slides. Maybe I should do the same. Varun, do you mind just telling us what you understand about it? I know that you said you've got a rudimentary understanding, but just give it a go, because it will help you find out what you don't know and what you would like to know. Yeah, good. You've got like one of the points about SNPs. Now, when you see a fancy word in biology, you should pause and break down the meaning of that word. Single nucleotide. What is a nucleotide, Ria? I'm waiting for 3 words. What makes up a nucleotide?
It's a part of DNA. It's like the part that connects to the
2 things.
sugar phosphate, deoxyribose.
Deoxyribose sugar. That is a type of sugar, right? And there's a phosphate group. DNA is a repeating unit of 3 things. You forgot the most important thing. The base. There is a base, A, T, G, or C, connected to a deoxyribose sugar. Why is it called that? Because a type of sugar is ribose. Has anyone heard of Ribena? That little purple drink? Okay, well I think that has ribose sugar as well. It's just a type of sugar. And it's deoxy because it lacks an oxygen. It's easy, right? And that's connected to a phosphate. If you do chemistry, what's phosphate? Come on. Here for 3 minutes, right? If you do biology, wipe this from your brain. Good. So that is a phosphate. So base, sugar, phosphate. That is a repeating unit of DNA. So it's called a nucleotide. You need to know that. Single nucleotide. We're talking about something at the level of 1 nucleotide. Everyone with me, right? Polymorph. What does poly mean? What does morph mean? Good. Many variations. So single nucleotide polymorphism means that at the level of 1 base, there are variations. Well, of course, you all learned that. What are the 4 variations you can have? An A, a T, a G, and a C. A fancy way of saying there are many variations at the level of 1 base. Using this word here. You all knew it. We're just labeling it. You'll agree, some of us might have an A at our first ever base on chromosome 1. Some of you might have a T. Some of you might have a G. Some of you might have a C. We would point at that base or that region and say that is a region where there is a single nucleotide polymorphism. Pause for a moment. What this means is that there are regions of DNA where there is no polymorphisms of a single base. In reality, I want you to understand this. Are you smarter than a chimpanzee? Yeah, exactly, right? Hope so, right? You're studying biology, they don't understand it. Now, do you know what percentage of your DNA is identical to a chimpanzee's? 98.5% is similar. That 1.5% enables you to have opposing thumb knowledge, to be cognizant of the world, the universe, your position, have existential crises, right? That 1.5%. Now, think about you and the person next to you. If a chimp and you are only 1.5% different, you and the person next to you are literally 99.9% the exact same DNA sequence. And in reality, what this means is if I get a strand of DNA, everyone on this planet has the exact same sequence for about 300 base pairs. And then at every 300th base pair, this is approximate, right? Everyone on Earth will have some kind of variation. 20% of the population will have a C. 10% will have a T. Some of you will have a G. Some of you will have an A. Does that make sense? If I read your genetic code, all of us have literally the exact same sequence, but very rarely some of us will have a different letter. When that happens, we circle it, we point at it, and we call it a single nucleotide polymorphism. It makes sense. There is a variation of one base in this population. Yes? Does this apply to? Depends what kind of twin we're talking about, right? There's monozygotic and dizygotic twins. We'll come back to that because I need to explain what the different types is and I'll go away from SNPs. So remind me after SNPs, I'll explain that. Does everyone understand that? Every 300 bases, there will be a variation of one base in the human population. We call that a single nucleotide polymorphism. We should just call it a base variation because that's exactly what it is. Now, there's something very important from an exam point of view about the word SNPs. SNPs have a very important point in the definition, and I've bolded it for you. Do you all see this here? If you don't write this, you will not get a single mark in your explanation of a SNP. When we were discovering variations of DNA, we needed to know which variations were significant enough to study and which ones that were just too rare that we shouldn't care about them, right? So let's say we're looking at DNA. Let's say we're looking at, pick a chromosome number, anything. 25. Looking at the 25th chromosome, and at the 307th base, everyone on Earth, 99.9% of people have an A, and 0.1% of people have a T. Is it worth studying, documenting, and looking at variations that are associated with disease? Probably not, because there's so much information in the genome. But if I go to the 670th base variation and I see 32% have an A and 70% have a T, well, that's a significant variation. There's pretty much a binary in this population. We should study this. We should call this a SNP. The definition of a SNP also depends on proportions. More than 1% of the population needs to have a variation. If less than 1% of the population has a variation, we don't call it a SNP. It's just a variation in the genetic code. Does that make sense? Okay? From an exam point of view, this is the most important thing. You don't say it, you're not going to. Good. Any questions so far? Okay. Very good. Now, this is another very important point. Where do SNPs come from? Where did we get these variations in our genetic code? Now, I want you all to remember this, right? You all learned about meiosis, fertilization. What happens in meiosis and fertilization? Do we make new letters? Do we change a capital R to a little r? We simply just change their organization. They move to opposite ends of the cell. They do this intricate dance where they end up in a gamete, and then they fertilize. The only way you can make a new letter, right, change a little r to a capital R, or make a completely new allele is through mutation, right? How do we get mutations, everyone? There are two ways. Errors of DNA replication, so spontaneous mutations that happen in your own cells just because copying 2 meters of A's, T's, B's, and C's is pretty hard. You make mistakes sometimes. What is the other way you can get a mutation? Pardon? Yeah, so that's again a bit of like, no, you won't get a mutation in crossing over because you're not copying anything, you're just swapping things around. That's just creating variation. Yeah, right? Anyone, I've seen so many stories of people going out to the beach or to the Great Barrier Reef because that's a really good holiday hotspot in this time, right? The sun, UV radiation, is a major source of mutation as well, right? So mutations, I want you to just put 2 things for now because the whole next module is about mutation. They can be spontaneous, aka arise from errors in DNA replication, or they can be induced. And this is through what we call mutagens. Mutagens. Okay. Good. All right. Any questions so far, Adam? When I first learnt about SNPs, I found it a bit tough to understand, but I think it's going pretty easy for you guys so far. Okay, good. All right, now I'm going to give you all a very tough question. Don't look at your notes. Why are SNPs more prevalent in non-coding DNA? This is a question I'm going to leave you all with, and I'll answer it at the end of today, which is in 3 minutes. Why are SNPs more prevalent in non-coding DNA? Feel free to discuss with your peers. Make sure, because you'll maybe close your books. Don't even let yourself read, because you miss out on a huge learning opportunity when you just read and try memorizing this. Sorry, I'm just going to reshare the screen for all of you. Give it 1 sec. Brooklyn, can you see the screen again?
Yep.
Can you see the text on the screen? Or is it just
Yep.
the Teams?
No, I can see everything. It's okay.
Okay. So it's just the internet of mind. Okay, good. Gayatri, Harshal, Raya, any questions at all so far?
No, I'm good.
Me too.
Good. Good. All right. Okay. And guys, yeah, feel free to bump in, ask questions whenever, okay? Just hit the raise hand icon and I'll answer it. Yes? Yeah, and what does that mean? But you'd still call it a SNP. You're getting there, think about it. Have you learned about SNPs at school? Yeah. Okay, well, I think the one thing created is it says use thought experiments. What would happen to a SNP? Guys, anyone got an answer yet? Okay. I want you to think about this. What would happen to a SNP if it was encoding DNA? What would happen to a SNP if it was non-coding DNA, right? That's what this is all about, right? What I'm trying to get you all to do is use thought experiments, right? Generally in life, no matter what field you go into, whether it's science, whether it's business, whether it's commerce, making an export-import decision, use thought experiments. If you follow through with that scenario, what happens after that? I think that's what intelligence is. It's not how much you can memorize HSC Bio. Now, if it's more prevalent in coding DNA, what would that mean, everyone? So let's say we've got a SNP in coding DNA. What does that mean? What does coding DNA mean to all of us? Good, right? So if you have a SNP in coding DNA, it's going to be expressed as a protein, and that's going to affect phenotype. Does everyone agree? Now, SNP means variation. Do you all agree? So if there's a variation in a base, there'll be variation in the protein, there'll be variation in a phenotype. Pick a phenotype, any human trait. Height, it's the height, right? I like to use this thought experiment a lot. Let's say that in this environment, all the food is on the ceiling, right? There'll be people with a variation. There'll be short people, there'll be tall people, right? As a SNP results in variation, the phenotype will be varied. There'll be tall and short people. What happens to all the short people? And so what's going to happen then to the variation of that particular base? The unfavorable base will be removed from the gene pool until less than 1% of the population have it. And the favorable allele, whether it be a T instead of a G, will be propagated because those organisms survive, reproduce, pass it on until more than 99% of the population have it. Can we call it a SNP if the variation exists in less than 1% of the population? There you go. That is why. The answer is SNPs encoding DNA are acted on by natural selection, which changes their frequency until less than 1% have an unfavorable variation. Right? SNPs in non-coding DNA never get converted into protein, never get expressed as phenotype, and thus they can remain hidden in the human genome. Make sense? My T and your A in our 603rd base position on chromosome 10 is not going to affect our survival likelihood because it will never make a protein.